Welcome everybody to today's edition of our webinar on emerging topics in biomolecular magnetic resonance. So we have two fantastic speakers today. So I'm really looking forward to it, and both agreed to recording of the lectures and uploading to the ISMRBS YouTube channel. I also remind everybody, feel free to put your questions into the Q&A box, either during the talk or at the end of the talk, and then we have a short Q&A session after each talk to address these questions. And very importantly, we have the informal part afterwards where we can have more extended discussions about the talks. So thank you very much for being here today, and I hand over to Christina Redfield, which will introduce the first speaker.
Right, it's my pleasure this afternoon in England to introduce Jeff Hoch. So Jeff studied chemistry as an undergraduate at Boston University. Jeff stayed in the Boston area to complete an MA and a PhD in physical chemistry at Harvard University. I first met Jeff more than 40 years ago in 1980 when we were both graduate students in Christopson's group at Harvard. Jeff was working jointly with Christopson and with Martin Karplus, and the latter two continued to collaborate over many decades, I think, as a result of being working together with Jeff at Harvard. Many in the Dobson group benefited from very useful computer programs that Jeff wrote, which provided things like lists of protons in close proximity to each other based on the crystal structure, which we then used to analyze 1D NOE spectra. He also wrote a great program that calculated chemical shifts, and in particular ring current shifts, which in those days were incredibly useful for our early efforts to assign the NMR spectrum of lysozyme. And actually the computer programs that Jeff wrote really motivated me to learn some Fortran programming, which I've used ever since in writing little programs to help in my own data analysis. So these are a couple of pictures from the Harvard days taken between 1979 and 1981. The picture on the left features, I'm not in that one, but it features Jeff and various other group members. So there's Jeff, Fleming Poulsen, David States, you'll all know the name from States TPPI, Randy Badin, Muriel Delapierre, who may be in the audience today, Mark Wagman, and Ed Olenichek. And then I joined the group in 1980, so I'm here featured in this. We've all turned a little bit grayer since 1980, but actually it was a great environment in which to work, and many of us stayed in contact over the years. So I moved with Christopson to Oxford in 1980, along with Muriel Delapierre, but Jeff and the others who were further along in their PhD stayed at Harvard. So despite the fact that Jeff and I were on different continents, we stayed in contact and have been good NMR colleagues and also good friends ever since then. After completing his PhD at Harvard, Jeff moved to the newly established Rowland Institute for Science, which had been set up by Edwin Land of Polaroid fame. And this was located in a fabulous site on the Charles River overlooking Boston. Jeff had a JL spectrometer there, and he continued to work on biomolecular NMR, but also in developing a wide-ranging expertise in many computational aspects of NMR. He worked on NMR data processing and developed the Rowland NMR toolkit. He had an interest in maximum entropy and other non-Fourier spectrum analysis methods. He considered the question of accuracy versus precision in NMR structures, and worked in the area of automated assignment. I visited Jeff at the Rowland Institute many times, and we worked on a lot of ideas together, often over coffee and donuts overlooking the Charles River. At Rowland, Jeff worked closely with Alan Stern, and he also collaborated with Gerhard Bogner's group at the Harvard Medical School. In 1990, while at Rowland, Jeff, Fleming Poulsen and I organized a NATO advanced research workshop on computational aspects of the study of biological macromolecules by NMR, which was held at Il Ciocco near Pisa in Italy. And that wonderful meeting was the precursor for the Gordon Research Conference on computational aspects of biomolecular NMR that many of you will have attended over the years, often held at Il Ciocco. So we've all grayed a bit over the years, and some people have lost a bit of hair. This is a picture much later in 2006, on the occasion of Fleming Poulsen's 60th birthday. His group organized a symposium to celebrate this in Copenhagen, and some of us traveled from abroad as sort of made guest appearances at the symposium. So here's Muriel, Chris Dobson, Fleming Poulsen, Jeff, and me. Sadly, we've lost Fleming Poulsen and also Chris Dobson, but Muriel, Jeff, and I are still hanging in there. Jeff moved to the Department of Molecular Biology and Biophysics at UConn Health in Farmington, Connecticut in 2004. And there he's the director of the Gregory Mullen NMR Structural Biology Facility. Jeff has continued his joint research interests in both experimental and computational aspects of biomolecular NMR. His NMR Box platform, which provides easy access to most of the software used by the NMR community, has provided an enormous benefit to all of us. Jeff is also serving our community through his leadership of BioMagRes Data Bank, the BMRB, which moved from Madison to UConn following John Markley's retirement. It's my pleasure to introduce him this afternoon, and he'll be talking about back to basics, how NMR can help structural biology navigate the era of big data and machine learning. Over to you, Jeff.
Can you hear me now? Great. Christina, thank you so much for that. One interesting little, I want to get those pictures from you, by the way, Christina. The one that you showed that you are in, that was on the right, that an interesting tidbit about that laboratory is that it's the lab where Fieser and Fieser developed napalm. With that, let me start sharing here. Okay, what do you see the title slide? Okay, excellent. So I'm going to talk about some topics that will be familiar to virtually all of you, and mostly not new ideas, but there's some ideas that have been given new currency and import, I think, because of recent developments in machine learning and big data. And of course, if you've been aware sentient at all over the past 12 months or so, you're well aware of really the phenomenal developments that have come in the world of machine prediction of protein structures from sequence alone, with in particular the efforts of the Google DeepMind team with AlphaFold and some also very impressive efforts coming out of the Rosetta team with RosettaFold. So my hope is that it'll stimulate a lot of strong opinions and discussions because of your familiarity with many of the things I'm going to talk about today. Let me just briefly acknowledge that this is work from a really phenomenal team here at UConn Health, but also some fabulous collaborators. I will mention individual names at various times rather than run through them all here. But in particular, I want to thank Joseph Courtney, Kylie Walters, and Guy Monteleone for some very helpful discussions and some data in advance of publication that I've made use of today. So quick outline, a bio-NMR, first off, really doesn't qualify as big data, although I think we, in principle, could get there soon. I'll talk about how the path to getting to big data, the machine learning revolution that we're dealing with, some ideas from Bayes and Haldane about how to deal with some of the challenges that validating predicted structures pose, and I'll close with an NMR-inspired approach to structure validation of structured predictions. So, what is big data? Well, it's a little bit like the Supreme Court in the United States definition of pornography. You know it when you see it. It's a little harder to actually give a precise definition. It's generally characterized by either by volume, the speed at which it gets emitted, the variety. But the bottom line is big data usually refers to data that's too big or complex for conventional software. the reason that's kind of a moving target is software gets more and more powerful, of course, so that what we term conventional software keeps evolving as well. So the goalposts for what constitutes big data keep moving as well. But the obvious examples were things like the Human Genome Project, the ENCODE project, electronic or health records, or EMR, electronic medical records. In the US, you will probably have heard of something called Epic, which is sort of taking over the electronic medical record market. And incidentally, the NMR community has had to compete with Epic for hiring programmers because they're based in Madison, Wisconsin. Google Images is a prime example as well of big data. And as my colleague Dave Donahoe likes to point out, the reason we've had a lot of the advances in machine learning for analysis of images is because of the billions of people who now have smartphones around the world and are constantly uploading image to the cloud. Just to mention briefly some of the technologies that have evolved to develop and manage big data. You will have heard of things like MapReduce and Hadoop, and more recent implementations like Spark for dealing with very big and very diverse data in distributed platforms. Well, to give you a sense of why BioNMR is not yet big data, there's just a snapshot of the content of BMRB. Incidentally, I want to give a shout out to John Markley and Eldon Ulrich for having conceived and built and been faithful stewards of BMRB for close to four decades. We are all really in their debt for having the prescience to forge ahead back when they did to establish BMRB. And I don't think we can really thank them enough for all the contributions that have evolved from that effort over the years. So as Christina mentioned, it moved to Yukon on the event of John's retirement. Eldon is now also retired, but I'm happy to say that they both are still involved behind the scenes and giving us the benefit of their insights on a lot of challenges that we face on an everyday basis. Very soon you will notice some changes to the BMRB website. It's being refreshed. One of the things that's happening is we are changing the logo. On the left is the old logo, and I got tired of being ribbed by some of our PDB colleagues about the use of the left-handed helix in the BMRB logo. So the new one on the right now has made a nod to right-handed helix that you find in biology. Let me just point out, although we've got an abundance of data in BMRB, it really still doesn't constitute big data, because it's something that really doesn't require a lot to store it. And the challenge is, how do we get more and more data in there? In particular, look at that number on the number of experimental data sets. That's really quite small. We really would love to be able to start hosting a lot more time domain data associated with the assigned chemical shifts that make up the majority of the depositions in BMRB. Well, BMRB, of course, has been a useful platform for developments that virtually all of us use on a regular basis. Things like Talos, the chemical shift index, Sparta, and all the various iterations of both Talos and Sparta really were enabled by BMRB. So one of the things, though, that on the path to big data is that the content of BMRB is growing to a point where we can now begin to use it in new ways. So Talos, Sparta, and those applications that are pretty routine now in BioNMR labs were largely based on general trends that were easy to discern from BMRB. Basically just sort of looking at the central tendencies of the broad distributions. But as the content of BMRB grows, things that we used to just dismiss as outliers are now beginning to be sufficiently abundant that we no longer can need to ignore them. We can actually start mining some of those outliers to potentially gain new insights. So there are enough of what we used to call outliers and ignore to now assign some statistical weight to surveys of investigations of what those outliers might be telling us. And I'm going to tell you just a brief story here about one such investigation. So back in 1988, Michael Levitt and Max Puritz posited that hydrogen bonds could form between an amide NH and an aromatic ring. They did an extensive theoretical survey of the quantum mechanical calculations of the potential energy of that interaction, found that it in principle could contribute about 3.5 kilocalories per mole worth of stability, and they pointed to one high resolution crystal structure at the time that suggested that this indeed can be found. There were subsequent surveys about a decade later of high resolution crystal structures by people like Stephen Burley and Greg Petsko that found anecdotal evidence of these in high resolution structures and counted several dozen putative instances of a hydrogen bond between an amide NH group and an aromatic ring. Well, Angela Gronenborn came to us with an interesting question a couple of years ago, and said she had this anti-HIV lectin that you've heard her talk about many times, had two amide NH proton resonances that were shifted upfield by rather extreme amounts, and she asked, could these be explained by ring current shifts? And the quick answer was yes, they're more than likely the result of ring current shifts, but that raised the question, could they also potentially be engaged in hydrogen bonds to those nearby aromatic rings? And so one of the things that we that Angela's question elicited was, can we look through those outliers in the BMRB shift data to find additional evidence, potential evidence of hydrogen bonds in proteins? So let's take a quick look at the distribution of amide proton shifts. The top row here is just the z-score, which is just the difference from the mean of amide chemical shift in units of standard deviations, and a negative score here is an upfield shift. A positive score is a downfield shift. So the top row is just looking at the distribution of the chemical shifts as a function of the distance to the nearest aromatic ring. So the first thing you notice is, of course, is that there's this relatively strong correlation that the larger deviations, that is the larger magnitude z-scores, do indeed correlate with proximity to a nearby aromatic ring. The bottom row is just zoom in on the early part, the part of that, the short distance information, and we can get a little bit better look at the distribution of those deviations. And you'll notice that there's some subtle differences there in terms of the outliers. In particular, histidine tends to have more downfield outliers, and the other three aromatic rings tend to give rise to more upfield outliers. To take another closer look, and again, this is a federation of the data in the BMRB with the corresponding structures in the PDB. Here we can take a look at what we're calling the azimuth angle, which is just the angle between a vector aligned with the center of the ring, normal to the ring plane, and the angle formed by that vector relative to the vector between the ring center and the amide protons. So if it's directly above the ring, the azimuth angle would be 0. If it's in the ring plane, this azimuth angle would be 90 degrees. So it's interesting that we do see peaks in this distribution, but what's interesting, in particular, if you look at for example, in the case of tyrosine, the peak in the distribution doesn't correspond to being directly above the aromatic ring. And I have to thank a reviewer on a manuscript that we submitted on this for asking us, why is it at around the first peak in that distribution around 25 degrees? Turns out that 25 degrees is the angle that you would get if the proton, the amide proton is NH vector is pointing towards the ring, but is not aligned above the ring center. It's aligned above one of the atoms in the aromatic ring, which is quite interesting. So it gives new insight into a bit of a tweak to the hypothesis of Perutz and Levitt that the hydrogen bond would form favorably pointing to the center of the aromatic ring. Instead, it's actually pointing directly at some of the p pi electrons of the aromatic ring, which makes a certain amount of sense. So just quickly to summarize our findings, we found that the similar distributions for phenylalanine and tyrosine for upfield and downfield shifts. For tryptophan, we found that mainly upfield shifts, and for histidine, mainly downfield shifts. And so in the case of histidine, it appears that it's more common for amide NHs to hydrogen bond to one of the electronegative nitrogen atoms of the histidine ring rather than to the p pi cloud. The peak in the upfield distribution near 30 degrees that we saw in the azimuthal angle, again, is consistent with the NHs being positioned, if they're involved in a hydrogen bond, above the directly above the p pi orbitals of the aromatic ring rather than above the center of the aromatic ring. And furthermore, the sheer number of instances we found by doing this federation of shift outliers in the BMRB and the structures in the PDB suggest that it's not anecdotal, that these kinds of hydrogen bonds are probably far more prevalent than people ever realized. So one of the take-home point I wanted to make is that the outlier number of outliers is getting to be pretty substantial, and there may be other opportunities for people to mine BMRB to gain new insights. So some possible examples, we might be able to look at shift equivalents for CHs, for example, to probe things like dynamics, potentially look at shift equivalents for opposite sides of the ring in the case of phenylalanine or tyrosine, for looking at phenylalanine and tyrosine dynamics. Your application here, I'm sure people in the audience will my hope is will think of other uses for some of these outliers, and to do that, to help people think about that and to actually do it, we've implemented something on the NMR box platform we're calling a Reboxitory, which is a data lake where we've created snapshots of the entire content of BMRB, the entire content of PDB, the entire NCBI non-redundant sequence database, and the entire AlphaFold structure prediction database, all as file system mounts on the NMR box platform. So you can do surveys by just pointing to the path of those things on there. We also snapshot them at regular intervals so that you can reproduce a survey, because as those databases continue to expand. But if you want a persistent and stable version, we've snapshotted them by time. We're doing it on roughly a quarterly basis, so that you can have some reproducibility of any surveys that you might do. So I encourage you to, those of you who have NMR box accounts, to point your browser over to look at the content there. And we're also open to suggestions for additional data repositories that we could post on that on the NMR box platform in the data lake. Another step towards big data I wanted to tell you about. This is something that Chad Rienstra was really originally scheduled to tell you about. I'm going to just give you a quick peek at it. It's something called the Network for Advanced NMR. It's something that's been launched on the basis of a $40 million mid-scale research infrastructure grant from the National Science Foundation. Some of the key features are it's going to result in two 1.1 gigahertz NMR spectrometers being installed for open access in the US. One devoted for biosolids is going to be at the University of Wisconsin-Madison, and one for solution NMR is going to be installed in Athens at the University of Georgia. Here in Connecticut, we're going to be building what's called the NAN Hub, which is going to be the cyber infrastructure for getting access. It will help with resource discovery, finding out what instruments might be best suited for a given problem. In addition to those 1.1 gigahertz NMR spectrometers, all the spectrometers at these three sites are going to be connected to this hub. Once fully operational, we'll have about 20 instruments connected. All of them will be streaming their data to a secure repository here in Connecticut on nearly real-time basis, and will make it very easy for investigators to push those into public data repositories as well. In addition, we'll be implementing to the resource discovery will help with scheduling, remote access. We'll be providing knowledge bases with help for standardized pulse sequences and protocols for sample preparation and analysis workflows. And again, the co-PIs on that are Katie Hensler-Wildman and Chad Rienstra from the University of Wisconsin, and Art Edison from the University of Georgia. So just to start to wrap up, again, the machine learning revolution is here. We saw an early evidence that it was coming back in 2018 when AlphaFold won the CASP13 competition. But what's interesting is that although there were headlines at the time that AlphaFold had won, I don't think it was fully appreciated at the time how badly it was really beating the human competition. So I reanalyzed on the right here some of the results from CASP13, just to show you just how much better AlphaFold was doing than the nearest competitor. So that if you look at these are the GDT_TS as the aggregate score that was used to rank order the results in the CASP competition, and with higher numbers being better. And what I've plotted here is the difference between the illustrating the best score versus the second best. And in all of the instances where the delta was very large, it was AlphaFold that was winning the day. And again, I want to thank Guy Monteleone for not only giving me access to the raw CASP results, but also helping me navigate that data. So we had this early inkling that things were about to change, and of course, this past year, with now AlphaFold now making stunning predictions on a proteome-wide basis for crystal structures, the day is here. So a lot of labs are now mining the AlphaFold database to get a sense of how well it's doing. And I would say the results are mixed. While there's some astonishingly good structures there, there's some things that AlphaFold noticeably hasn't done terribly well. What intriguing, Guy's group has analyzed the AlphaFold structures that were used in the CASP14 competition, and found that some of the AlphaFold structures are actually better than the deposited structures corresponding to the experimental NMR data, which is kind of an amazing result. Contrast that with Kylie Walters' findings that while AlphaFold does pretty well in case of a protein that she's looked at extensively, while it mimics one of the members of the NMR ensemble, it really doesn't capture the overall features of the NMR ensemble, which suggests that maybe one of the things that which isn't surprising, that AlphaFold isn't quite yet getting insights into the nature of dynamic proteins. And Mike Williamson's group has got a preprint out finding And they find essentially what Guy and Kylie found, that is kind of a mixed bag. There's some astonishing successes in the AlphaFold prediction database, but there are also some instances where it doesn't do quite so well. So I think and I know that there are other groups. I know AdBax's group is working extensively on this now too. And I think we're going to see some really exciting results helping us understand both the strengths and limitations of the AlphaFold approach to predicting structures. So I just want to wrap up here with a couple slides about an approach for how to validate machine language predicted structures based on Bayesian analysis, but it's kind of also based on a kind of a really astonishing essay that was written by J.B.S. Haldane. You guys will remember he was a biochemist. I believe there's a kinetics equation named after him. Art probably will know it better than I do. But he's got this really amusing but very insightful essay on how to fake scientific data, and how difficult it would be to avoid detection if somebody were to analyze the distribution of errors in your prediction. And so it turns out that detecting a fake is really very similar and essentially isomorphic with validating a structure. A good fake, like a good model, is going to essentially be indistinguishable from the truth. And so Haldane's procedure for detecting fakes is really also a really good proposal, a good approach to validating a predicted structure. So the basic idea is that the calculated properties should be comparable to the experimental values. The distribution of errors should be comparable in magnitude, should be comparable to the experimental uncertainty, chi-squared, but the higher order characteristics too, the deviations should be properly distributed. So while that's Haldane's prescription for detecting a fake, it's also a useful strategy for validating a predicted structure to see how well they are in concordance with any empirical data. And so that raises the question, our NMR structures, how are the errors distributed? And we can mine the BMRB to look at this question. Here I've done a small calculation on a data set that was used by, developed by the Northeast Structural Genomics Consortium. And I thank Guy Monteleone for this database. It's about 36 structures that happen to have both high resolution X-ray structures and NMR structures. And so in this case, I've plotted the distribution of deviations of the model from the NMR distance bounds that are deposited in the BMRB. And if you look broadly at this, it looks like it might be a reasonable error distribution, except for one thing. There's this spike there near zero that's kind of unusual, and the source of that really is no surprise. It really is simply the result of the fact that most of the structures in NMR structures in BMRB and PDB use a flat well restraint potential, which applies no force to the structure if the distance falls between the upper and lower distance bound. But then as soon as it exceeds the upper bound, applies a quadratic force, restoring force. And so that peak in the distribution is just the result of the fictitious force at the boundary. So that's not something that you would expect of a realistic distribution of errors. So it would be pretty easy to based on that, to say we're not doing a terribly good job in a Bayesian sense of making sure that, or Haldane sense, that our distribution of errors mirrors a realistic distribution. A very simple solution is to, instead of using a flat well potential, is to use something that doesn't give rise to a fictitious force at the boundaries. So one way to do it is to simply just put a restraint on the calculated NOE value rather than on a distance, and you get a smoother distribution, as you see in the bottom here. This basic idea is, let me jump ahead here since I'm running a little long, isn't terribly new. It's been around since the early 1980s, and it actually was something that Jim Prestegard and Horst Kessler proposed in dealing with using scalar couplings to restrain protein structures. Their motivation was largely to, instead of restraining dihedral angles, to restrain calculated J values, was to incorporate the inherent ambiguity in the Karplus equation. But it turns out that it also is a way of ensuring that the distribution of structures mirrors the experimental uncertainty. That basic idea could be then generalized to the notion of Bayesian restraints, so that you're putting a quadratic restraint on a forward modeled or a back calculated observable rather than on dihedral angles or distances. So in other words, restrain J values, restrain calculated NOEs instead of restraining distances and angles. The general solution to this was elegantly posed by Michael Nilges and his group back in 2005. It's something called inferential structure determination, which is really a very rigorous way of solving this problem. The only issue with inferential structure determination is because it makes no assumption about the nature of the error distribution, it requires extensive exhaustive sampling in order to map out the distribution of errors. So as a result, it's very costly to apply. But later on, Michael and his group realized that this Bayesian restraint approach is a less costly way to get results that are very, very, very similar to what you would get from a more costly inferential structure determination approach. Okay, in the interest of time, I'm just going to jump ahead and skip a couple slides, but just leave you with this proposal for a workflow for structure validation of machine learning predicted structures that basically would just start with a workflow for taking a model and doing the forward modeling to predict experimental observables. The level of accuracy could vary depending on how accurate your model is, whether you're working with a coarse grain model at one level or an all atom model and explicit solvent, whether you're dealing with a single rigid structure or an ensemble, whether you're using an isolated spin pair approximation in the case of predicting NOEs versus a full-blown density matrix approach. If you're calculating structure factors for X-ray data, are you using isotropic B values or anisotropic, or are you using a more accurate second order expression for F-calc for the accuracy of the scattered wave approximation? In cryo-EM, are you using a full map, half map? So this isn't meant to be exhaustive, but just give you an idea that this kind of approach could be adjusted for the level of detail and accuracy of the forward modeling pipeline that you're using, and then basically use that forward modeling to calculate a prior probability in the Bayesian sense, how likely the structure is to agree with existing knowledge, while at the same time calculating a likelihood using Bayesian restraint functions in order to get a proper likelihood or chi-squared statistic. And it's important to do that in order to be able to combine likelihoods from different approaches. So for example, in the hybrid or integrative approach, you need to be able to do that in order to know how to weight NMR data versus, say, SACS data at the same time. In essence, the only way to do that with equanimity and in a consistent fashion is to weight them according to the experimental uncertainties of each of the approaches. So aside from this general approach, to validating machine learning structures, I think it's probably also likely to become a new workflow for instructional biology, that in many instances we're actually going to start with the predicted structure and start collecting our data to empirically validate an AlphaFold or Rosetta Fold structure, rather than doing de novo empirical structure determination the way we've been doing it for the last 3 or 4 decades. With that, I've run a little bit long, and I want to stop here and thank you, and I hope this will stimulate some strong opinions among the attendees here. Thanks so much.
Thanks, Jeff. Maybe I could read out a couple of questions for you from the Q&A box, and you can answer them. So first question's from Carol Post. She says, hey, Jeff, the outlier analysis is quite interesting. An immediate consequence of the potential aromatic hydrogen bond is the question of molecular dynamics force field modification. Any idea about the energy level that might be involved with these interactions, say relative to a typical hydrogen bond interaction?
Yeah, so we asked Milo Wessler to actually do some Gaussian calculations for us, and he more or less agrees with the results that Levitt and Perutz came up with, of about 3.5 kcals per mole. So that's important. I think that's significant enough to worry about. So as a starting point, I think we could probably use Gaussian calculations, Carol, to come up with a model for empirical term in say CHARM or AMBER that would try to capture this.
Maybe I can ask a follow-on question, again, related to these interesting hydrogen bonds to aromatic rings. Do you think the existence of these hydrogen bonds would have any consequences for aromatic ring flips? So say with a tyrosine, has anyone looked to see whether those tyrosine aren't flipping, say, in the way that a non-hydrogen bonded one would flip? And also, are there any consequences for the chemical shifts of the aromatic protons for those rings which have these hydrogen bonds?
Yeah, I sort of alluded to that, Christina. That's something that we're really intrigued by. Are the tyrosines or phenylalanines that are in slow exchange for flips about Chi2, do those tend to correlate with these hydrogen bonds? We've just started looking at that. We don't have an answer yet, but yes, I think that's the next interesting question to address. This issue of hydrogen bonds to histidines to the nitrogen may also have an echo in ring dynamics about Chi2. So it would be quite interesting we haven't looked at MD simulations yet to correlate the Chi2 fluctuations with the presence of these hydrogen bonds, but that would be a really interesting study to do.
Got a question from Andy Bird. Would you like to comment on the modification of your final workflow to use Bayesian tools for structure validation in the PDB depositions?
So yes. So what's interesting is I've had conversations with the other members of the WWPDB about this, and there's a concurrence among PDBE, PDBJ, and EMDB that we really need to look more carefully at improving the rigor of the PDB validation process. It's really essentially the same problem as that's faced with validating the machine learning structures. So we've got a lot of ad hoc and heuristic metrics that are in the PDB validation pipeline, but we really need to do better at bringing some statistical rigor to that validation. And so the PDB is going to be announcing pretty soon, I hope, a series of community-based workshops to try to establish a consensus on more rigorous statistical methods for validating things in the PDB structure validation pipeline, as well as validating predicted structures. So yeah, Andy, thanks for for letting me alert people to keep an eye out. Those are going to be the plan is to try to have those workshops on a 6-month basis, because things are happening very rapidly in the machine learning world, and the intent is to move them around the world so they will be conducted on an international basis, both in the US, the UK hosted by EMDB. We'll also hold one in Osaka hosted by PDBJ.
Okay, maybe just a final question before we move on to the next formal presentation. There will be a time at the very end in the less formal session for further questions, but I've got a final question from David Eliezer. Tyrosines are strongly implicated in weak interactions leading to liquid-liquid phase separation, in part based on pi-pi interactions. Do you think these hydrogen bonds could contribute to weak interactions in IDPs and or in liquid-liquid phase separation?
David, I really think that's something that needs to be looked at. Yeah, it occurred to me as soon as I saw this that this might be a significant a contribution to the aromatic interactions or the phase transitions with liquid phase separation. Question is how to go about it. You've studied this more extensively than I have, but one of the concerns that I have is that there's an unfortunate poverty of data for disordered proteins in BMRB. So I'd like to encourage you and your colleagues to try to get more NMR data on disordered proteins deposited. I think if we can build up the holdings in BMRB for disordered proteins, we'll have a better chance at addressing that question.
Okay, thanks for those questions, and Jeff, thanks for your responses. I think we'll now move on to the second speaker, and further questions for Jeff can be asked in the informal part of the meeting.
Welcome for the second talk. We are switching to one other magnetic resonance method, EPR, and I'm very happy to introduce Benesh Joseph, who is giving this presentation in the second talk. Benesh studied at Osaka University in Japan, and then for his PhD thesis he moved to the ETH Zurich and joined a group of Gunnar Jeschke and worked together with Enrica Bordignon. And with his PhD there, already working on biological application of EPR spectroscopy. After finishing his PhD thesis, 2014 in the ETH Zurich, he joined my group with a Marie Curie fellowship, which he got from the EU, and worked then together with me. And in 2019, he got a prestigious Eminöter Award from the German Research Society to build his own independent group. And then he switched from the physical chemistry department to the biophysics department, or the physics department with the Institute of Biophysics, where he is working now. He has his own group, his own spectrometers, and he will talk today about BAME, an outer membrane protein, doing pulse dipolar spectroscopy on that. Benesh, the screen is yours.
Thank you, Thomas. I hope you can hear me and also see my slides. So first of all, it's a great pleasure to share our recent research in this seminar series, and also thanks to Marcus for inviting us. So today I will briefly talk about the methodological aspects we have been doing over the past years to observe membrane proteins in their native environments, and the application of these approaches to observe BAME. So the world of in situ or in-cell NMR is larger and broader compared to the corresponding ESR field. So here, for example, in-cell NMR has been used to observe structure, dynamics, folding, maturation, chemical modification, drug interaction, etc. for proteins and nucleic acids. But in situ ESR spectroscopy or in general ESR spectroscopy, we are addressing dynamics of proteins and nucleic acids. And a definite advantage here is higher sensitivity and longer range of restraints that we can determine. So, most of the proteins are intrinsically diamagnetic. That means we have to introduce a spin label, usually through reaction of a thiosulfonate or maleimide functionalized spin label with an introduced cysteine. Of course, genetic engineering encoding is coming as an option, but it's still not at a stage to replace this labeling strategy. So although NMR has a wide repertoire of techniques, here we are mostly employing the double frequency technique called 4-pulse DR or PELDOR, for which a 5-pulse version is also available to determine even longer distances. And in some cases, single frequency techniques like double quantum coherence or rhythmic can be used, but their applications are limited, and the double frequency techniques remains as the method of choice. So over the past years, we have been working on extending the application of ESR spectroscopy to observe outer membrane proteins in their native environments. And indeed, other methods such as AFM, solid-state NMR, or cryo-electron tomography are also progressing in these directions. But in general, all these methods are challenged by lower resolution. So this is the schematic view of the cell envelope of a gram-negative bacterium consisting of an inner membrane and an outer membrane separated by the periplasm. So the outer membrane is an asymmetric bilayer consisting of phospholipids and lipopolysaccharides. And it also harbors numerous beta-barrel proteins, which are essential, some of which are essential for the survival. So when we talk about ESR spectroscopy of these proteins, in general, they are expressed at a very low level, about a few hundred to few 100,000 copies per cell. And for EPR spectroscopy, the stability of these spin labels in the cellular environment is a major concern. And also directly labeling the protein in the native environment make many challenges, including the undecided background labeling. So for EPR spectroscopy, nitroxide labels are the most popular one, followed by the gadolinium and the tritium labels. So MTSL is the most popular label, but it's very easily reduced in the cellular environment. And it has been shown that replacing these methyl groups with the diethyl groups can prolong their stability. And also the carbon-sulfur bond formed by maleimide groups are more stable than the disulfide bond formed by thiosulfonate functionalized spin labels. So coming to gadolinium and tritium, they are more bulky, as you can see from here, and also you can see the volume of the rotameric populations. Obviously, these labels occupies a larger volume. And here we tested the stability of these labels, except the gadolinium labels, in an ascorbate solution or E. coli suspension. And as you can see here, either tritium and the shielded nitroxide labels are very stable, but MTSL or POX, they're rapidly reduced in the ascorbate solution. And a similar behavior can be seen in the E. coli suspension also, where the POXL and MTSL is undergoing significant reduction over time in the E. coli suspension. Here we are talking about direct labeling of the cobalamin transporter B2B at the position 188 on the second extracellular loop. And the labeling is performed either directly in E. coli or isolated outer membranes. And as you can see here, all these labels give a much higher signal for the cysteine variant compared to the wild type. Indeed, we get a similar response when we go into the outer membranes. But the problem, not the problem, one observation here is that the background labeling is increased when we do the labeling in the membranes. So one particular thing here is that a definite advantage for EPR spectroscopy here is that the background labeling is random. And that means they specifically goes into the intermolecular part of the payload data, and they don't interfere with the distance discrimination, albeit reducing the overall sensitivity to some extent by lowering the effective modulation. So here we performed distance measurements between B2B and a spin label substrate called tembosine, which has a nitroxide spin label attached to that. So essentially, we labeled position 188 using these different labels and performed the distance measurements either in cells or outer membrane. So essentially, this become a nitroxide or nitroxide tridal or nitroxide gadolinium distance measurements depending on the nature of the label attached to here. So as you can see here, all these labels enabled except gadolinium. I will come to that later. So that all the nitroxide labels and the tridal labels enabled distance measurements in E. coli. And not going into the details, but a very interesting observation is that the overall spread of the distribution is not increasing corresponding to the size of the label. That means it's not the overall size of the label, but the absolute distance between the spin centers, which is most relevant for the observed distance distribution. So going to outer membranes, essentially we see the same distance distribution as we saw in the whole E. coli for the nitroxide and the tridal label. And in the outer membranes, we were able to use the gadolinium labels also, which didn't work in E. coli. But again, you see here that for gadolinium, there is a higher narrowing of the observed distance distribution, which also tell us that the physical-chemical properties of the tag itself, which can influence the interaction with the surroundings, also may influence the observed distance distribution. Okay, so in general, when we do a DIR measurements or payload measurements, the observable dipolar revolution time and as well as the longest measurable distances, this is mostly limited by the phase memory time, which is approximately the transversal relaxation time. So when we look into the echo decay here, we can see that the 4-pulse payload echo that decays at a rate very similar to the Han echo sequence here. And it is known that if we place these pulses under a corpuscle condition, where this second interpulse delay is approximately 2 times the first interpulse delay, this can minimize the effect of nuclear spin diffusion on electron spin relaxation. And as you can see here, the relaxation is significantly prolonged, and which could enable, in principle, the measurement of dipolar revolution time up to 14 microseconds, or distances up to 8 nanometers in the cellular environment. So, based on this corpuscle condition, a 5-pulse payload sequence was developed by Borbat et al., which required an additional pi pulse between the first 2 observed pulses to break the symmetry of the dipolar evolution around the primary echo. So a limitation of this pulse sequence is that these rectangular pulses have non-uniform inversion probability. That means a partial excitation 4-pulse artifact is overlaid on the primary data, and additional experiments or simulations are required to determine the distance distribution. So, in fact, in a paper itself, they suggested that replacing these pulses with broadband-shaped pulses, which have more uniform excitation within the bandwidth, can suppress this artifact. And that's what we did here. We replaced these pump pulses with broadband sectant pulses. And as you can see here, employing these pulses suppressed the level of artifact to a amount which has the least effect on the determined distance distributions in this case. So with this, I will switch to the system that we are working now. So, so the beta barrel assembly machinery complex is one of the most essential and conserved protein folding machinery in gram-negative bacteria. So essentially, the complex facilitates the folding of unfolded outer membrane proteins from the periplasm into the outer membrane. So this is a pentaheteroligomeric complex consisting of the central beta-barrel BAME, as you can see here, which interacts with the 4 other lipoproteins BAM, B, C, D, and E. And as you can see here, the complex has been, especially the central BAME barrel, which is the insertase here. This has been observed in 2 major conformations. One is an inward open conformation where the terminal beta strands, beta 1 and beta 16, they are zipped. And in the lateral open conformation, this terminal beta strands unzips, opening the barrel into the interior of the membrane. And this lateral opening further increases when a substrate is bound, which is the substrate show here. So despite the availability of several structures for this complex, how the system achieves folding and insertion of the protein into the outer membrane remains elusive. And most importantly, it is suggested that the native membrane environment as well as the symmetry of this environment might be very crucial for the function. So when we look into the conformations of BAME in the different structures, so I told you that there are 2 major conformations, a lateral open and the inward open conformation. So when we look into the lateral open conformation, we can see this in the BAM-ACD subcomplex, which lack the B subunit, or we can see it in the ABCD full complex in different detergent micellars or even same detergent micellars, or even in the nanodiscs. And when we look into the inward open conformation, BAME alone is observed exclusively in the inward open conformation. And if you look into the BAM-ABCD complex, this also has been observed in the inward open conformation, again in different detergent micellars or in nanodiscs. So what we see here is that there's no direct correlation between the conformation of BAME and the presence of lipoproteins or the nature of the surrounding environment. And all it suggests is that BAME is likely a very dynamic structure, and its conformation and dynamics could be modulated by the lipoproteins of the surrounding environment. So, so here to observe the conformational heterogeneity of BAME, we engineered cysteine substitutions at strategic positions along the BAME barrel. For example, position 501-796, this is incorporated between loop 3 and loop 8. And this loop 3 undergoes large conformational change, as you can see here, when the barrel transitions from inward open to lateral open state. So the 501-801 connects the beta 16 with the loop 3 again, and beta 16 strand is part of the lateral gate, which is also undergoing significant conformational changes. And then finally, on the lateral gate, we have this 434-801. This is position 434-801. This is exactly at the lateral gate on beta 16 and L1, which is connected to beta 1. And this would allow us to observe the conformation heterogeneity of the lateral gate directly. And going to the periplasmic side, we have 452-732. This is on this periplasmic turns, T1 and T6, 452-732, which comes closer when the barrel switches from the inward open to the outward open conformation. And finally, we have 359-780. This monitors the interaction between T7 and port of 5, and you can see that the port of 5 moves into the barrel when the barrel switches from the inward open to the lateral open conformation. So you can see the simulations for these positions on the inward open or lateral open state. And as you see here, these positions can clearly distinguish between these 2 major conformations of the barrel, namely inward open and lateral open states. And okay, so now here we went on for the purification and spin labeling of the BAME in endiomycetes, for example. So here you see the sec profile of spin labeled BAME, which comes out as a monodisperse peak, and we collected the pelro samples directly from this peak. And here you can see the SDS page of different doubly labeled cysteine variants, all of them elute with a high purity. And here also you can see that the cysteine substitutions doesn't affect the function of the BAME. So here, for example, with arabinols, the BAME is driven from the genome under the control of this arabinose promoter, and similar growth can be observed even in the absence of arabinose, where BAME is expressed, BAME variants, cysteine variants are expressed from the plasmid, which also supported growth to the same extent. So here you can see the continuous wave CWEPR spectrum with little or no free spins. And this also gave us a labeling efficiency of 70% or higher, which would enable us to obtain high quality pelro data. So as I shown earlier for the B2B, here also we went on for purification and spin labeling of BAME directly in the native membranes. So here you can see an SDS page of the BAME double cysteine or single cysteine variants. This is the band corresponding to BAME. As expected, there are other proteins because this is a native membrane preparation. And we further confirmed the presence of BAME by Western blotting for some of the variants, as you can see here. So a key question here, when you go for performing such experiments, the native membranes, is the expression level of the protein. Yeah. So here we performed spin labeling in the intact E. coli cells at position 501 on the loop 3. And you can see the background labeling for the cis-less variant here. And for the spin concentration that we obtained here, and from the number of cells in the sample, we estimated that we have about 300,000 of copies of BAME per cell expressed. So here you can see again the continuous wave CWEPR spectra without little or no free spins, and this is not an easy task to completely remove the free spins, which took quite some effort for Adira. And as I shown earlier for B2B, you can see that in membranes, we have a little bit higher background labeling. They are not coming from labeling of the proteins because outer membrane proteins doesn't have any free cysteines. But here, what happens is that the spin labels physically adsorbs to the membrane. But as I showed you earlier, as I'll show you in the coming slides also, they don't contribute any distances other than lowering the overall sensitivity. And so we have an effective labeling efficiency of 30 to 70%, which is also acceptable for performing measurements in the native membranes. So, so when we go for overexpression in the native membranes, one concern here is that does the protein dimerize, oligomerize, aggregates, and this would be very bad for pellet experiments. So here, first we measured instantaneous diffusion by decreasing the flip angle of these pi poles from pi to pi over 6. And as you can see here, there's no change in the relaxation as we reduce the flip angle, revealing that there is no high concentration, local concentration, as you would expect from oligomerization or aggregation. So we further measured several single cysteine variants, as you can see here, which is distributed along the barrel. And in all these cases, we see a monoexponential decay devoid of any distance, which rules out any dimerization or oligomerization. So this is an available structure of BAME where BAME dimer was stabilized by cross-linking. And based on this structure, this would be the distances we would expect for these single cysteine variants. And as I mentioned earlier, we don't see any such distances, suggesting that this structure is either transient or is not stable without cross-linking in the native environment. So now I would like to go directly into the measurements. So here you can see the primary data, the background corrected data, and the distance distributions. And the simulations for the inward open and lateral open states are shown in black and gray respectively. So when we look into L3, L8, we are measuring between L3 and L8. And as I mentioned earlier, L3 can undergo large conformational change. But here we see that the experimental data is mostly in agreement with the inward open conformation of the barrel. And we see a very similar response when we measure between L3 and beta 16. This is beta 16 and L3. And beta 16 is forming a lateral gate. There also the experimental data falls in agreement with the invert open conformation. And when looking directly at the lateral gate for 34801, there also we see that the experimental distance distribution is closer to the invert open conformation. And for the 452732, where we measure between these periplasmic turns, the distance is closer to the invert open conformation, but a little bit broader, which might be due to the flexibility of these turns. And when we look at the distance between port of 5 and T7, which is we are measuring between port of 5 and T7, this is the distance we are looking here. There also we see that the experimental distribution is closer to the invert open conformation, but a little bit broader than the simulations, again, likely due to the flexibility of the structural elements. So overall, what we can say is that in LDO-Mycellus, the BAME barrel has a conformation very similar to the invert open conformation. So now we went on to measure the native membranes, where we saw significant changes in the conformational heterogeneity. But here I would like to focus on three positions in particular. So for example, measuring between L3 and beta 16, we are measuring between L3 and beta 16. We see a very broad distance distribution spanning the range occupied by invert open and lateral open conformations. And when we look directly at the lateral gate for 34801, this is a functional hotspot of this insert phase. Again, we see a very broad distribution encompassing the distances corresponding to invert open, lateral open, and even lateral open substrate bound state, telling us that this region is likely very dynamic and conformationally heterogeneous when we observe the native membranes. And finally, when we come to the T7 port of 5 distance, where we measure between T7 and port of 5, you see that port of 5 moves into the barrel in the lateral open conformation. So one of the major distance is in agreement with the invert open conformation. And we see an even longer distance component, longer than this, suggesting an additional dynamics for P5, port of 5 domain in the native membranes. So one interesting observation here is that here we see distances corresponding to lateral open conformations. And if you look into the structures in the lateral open conformation, the port of 5 should move close to the barrel, shifting from invert open to the lateral open conformation. But here we don't see distances corresponding to the lateral open conformation for port of 5. And we speculate that this is likely because transducing the conformational changes of the barrel to the portal domain likely requires the lipoproteins, and we don't have lipoproteins in our sample. So possibly that indicates the functional relevance of the lipoproteins. So with that, I'm coming to the conclusions here. So when we observe BAME in LDO-Mycellus, BAME mostly exists in the invert open state, which is in agreement with the BAME structures. But when we go to native membranes, we see that BAME can sample different conformations, most importantly, without any of the lipoproteins. And that means the native environment, and most likely its asymmetry, has a significant influence on the dynamics of BAME. So here, one open question here is how the complex perform this function, how it fold and insert protein into the outer membrane. Of course, the exact mechanism remains elusive, but a consensus view on this mechanism is that a dynamic behavior of BAME might be the key for folding and insertion of the protein. And also this dynamic nature of the BAME might have an important role in destabilizing the membranes. So our observations on this of having a highly heterogeneous conformation of BAME is in agreement with a dynamic nature of the BAME in the native membranes. And this results also opens up the possibility to further look at the effect of lipoproteins on the conformational space, as well as the orientation, dynamics, and interaction of the protodomains with lipoproteins in the native environments. And in the long run, we hope that it will enable us to look into substrate folding and insertion in the native environment. And with this, I'm coming to the acknowledgement. So all the experiments on BAME has been done by Adita. She's a PhD student in the group. And most of the experiments on B2B I show you was performed by Sophie. She's a master's student in the group. And you know, all these beautiful spin labels, shielded nitroxide, myelimide functionalized triters, thisulfonate functionalized triters, they are synthesized and provided from Novosibirsk by Elena and Victor. And we are so happy about this collaboration, which has been running for a while now. And also I would like to thank David from University of Virginia. The collaboration with him in the beginning has been very, very helpful for establishing this methodology. And also I would like to thank Thomas for his continuous mentoring and support. And thank you. And also thanks to all the funding agencies. Thank you all for listening to my talk. And I'd be very happy to answer your questions.
Thank you very much, Benesh. Very nice presentation and very interesting results to compare the measurements in natural surrounding with more model membranes. So there are a number of questions. Let's start with that. From Andy Bird, there's a comment, a beautiful results. Can you comment on the use of nanodisk for reconstitution? Do you use glycerol in solution, deuterated glycerol, percentage, or something like that?
Yeah. So nanodisk we haven't used, but some groups are using it for cryo structure determination. And except one of the structures, all the structures in nanodisk were lateral open. So there is an idea now that probably the nanodisk environment promotes a lateral open conformation, but on the other side, we have one structure in nanodisk which is inward open. So, definitely that's something we want to check. We also are interested in trying different lipid compositions, different lipid thickness, for example, because the membrane properties are suggested to be a key which controls bump function. And glycerol we use here in our pellet samples, 10 to 20% deuterated glycerol we use, but this is present in both detergents and membranes. So I don't expect a selective difference due to glycerol because this is present in both samples. I hope I addressed your concerns. Yeah.
There is a question on the more technical side, but also important, of course, for the sample constitution from Gunnar Jeschke. With single labeled samples, you see a dimension below 2 for the intermolecular background function. Do you have an idea why and how do you fit background to extract it from the intramolecular distance distributions?
Yeah. So, so the exact dimensionality in these membranes is a little bit difficult problem to determine exactly, and it goes below 2. That's true, and I think this may be due to inhomogeneous distribution of the proteins in the membranes. It's a 2D membrane, but again, we don't know if the proteins are homogeneously distributed there. So this is a difficult problem to exactly determine, and I think this partial dimensionality around 1.1, 1.7 is probably due to this inhomogeneous distribution. So we experimentally fit the data from single cysteine variants into a stretched exponential decay by fixing the value of the dimensionality for spin distribution free, and that's how we see what is the nature of this exponential decay in the membranes. And we see that all the single cysteine variants consistently fit into a very similar stretched exponential decay. So, and then we used this kind of stretched exponential decay background for analyzing the data for double cysteine variants.
There is a further question from Andy Bird. Is there lipid dependence on the conformational distributions observed in micelles or nanodisk preparation compared to the natural membrane?
Yeah. So at the moment, we have between detergent micelles and native membranes. Here we see definitely a difference in the conformational dynamics. Yeah. So this was very exciting. Like in detergent micelles and LDO micelles, what was very interesting for us also is that in LDO micelles, BAME was crystallized in inward open conformation, and that's what we exactly see when we do the distance measurements. It's in the inward open conformation. But when we go to the outer membranes, the conformational heterogeneity is significantly diverse. And this heterogeneity falls very well in line with other conformations of the BAME. So it gave us a very affirming observation that the native membranes can modulate the conformational space. So I assume that this modulation of the conformational space could be lipid dependent or even detergent dependent, but definitely we need to do more measurements in these directions to make a conclusive statement. Yeah.
A question from my side. You have seen different degrees of labeling efficiency on different sites in the BAM-E and on the polymer proteins. Is there a kind of systematic? Is there some systematic variation that you have a higher efficiency on the outside of the cells if you do this in E. coli cells, and lower if you go further into the channel?
So that's a very interesting question. Actually, so the labeling efficiency doesn't really correlate with what we predict or simulate based on the solvent accessibility from the structures. So that means the accessibility is considerably modulated or altered by the cellular environment. So for example, some extracellular loops where we expect perfect labeling based on simulations, we don't get any labeling. And this is in fact puzzling us for quite some time when we try to do the labeling in the cellular environment. A lot of this exposed extracellular loops doesn't give any labeling. So I think the structure or the accessibility or the local structure and accessibility of these positions might be very different when we go into the native membrane or cellular environment. So we don't have any statistics for labeling efficiency moving along the protein. Yeah.
Good. I see no further question. Maybe something interesting for I've seen there is quite a number of EPR people listening to the talk, but of course there's a larger NMR community. Maybe for information, Benesh, how big is your sample volume for this? If you take the E. coli cells, how much volume do you have?
So these measurements, in general, we have about 10 microliter sample in the resonator. So the active volume is even smaller than that actually. So it's about 5 to 7 microliter. That's what we use for the measurement. So I would say it's a very sensitive experiment, yeah. And so we have this expression level of about 10 to the power of 100,000 copies per cell, yeah. And that's the reason the expression level became a key factor to do a successful measurement. But the sample volume is very, very small from that perspective. Yeah.
Okay, thank you very much. I think we answered all the questions.
So
thank you very much.
Thank you very much.
Everybody.
Yeah.